the Science Guy.
Hello guys, I'm Mr. Nelson. Today I'm going to be talking about eukaryotes and gene expression. Okay, let's start with the question, why do eukaryotes have to have an on and off switch? It's because an organism such as us needs different gene products at different times in its life at different places in its body. For instance, what your body needs when you're an infant is different from what your body needs at a later stage in life. Let's start out with 5 levels of control. Oh, whoa, let's delete that. Level 1, DNA access. This controls what genes are accessible. These are histones and its histone tails. That's the DNA tightly coiled around it, and those are amino acids. This is a whole nucleosome. These are multiple histones wrapped tightly coiled by DNA chromosomes, and because they are so tightly coiled, no access is available to the DNA or proteins, meaning none of the information is going to be available to the RNA polymerase to transcribe the information within the histones. So these are histones, right? And when acetyl groups are added to the histone tails, it begins to loosen up these tightly packed histones. Because they're becoming relaxed and not as tightly packed, the DNA is actually accessible now. So the tightly packed ones are called unacetylated, while the looser ones are called acetylated. The unacetylated histones are also known as heterochromatin. Note the prefix. This means no transcription is allowed. Acetylated is also known as euchromatin. This means transcription is allowed. DNA methylation is, what is that? When they are loosely packed, that means it is unmethylated. While when it's tighter packed, it's methylated. DNA methylation is when methyl groups are added. Level 2, pre-transcription. Necessary mechanisms are required to begin the process. Eukaryotic genes work with a lot of upstream regulatory elements such as enhancers and proximal control elements. Upstreams are DNA sequences that comes before a transcription unit that needs to have specific proteins present for RNA polymerase to begin transcription. Proteins that control RNA polymerase are known as transcription factors. Proteins work with other proteins to get the RNA polymerase to bind to promoter to begin transcription. Controlled transcription is a major way for cells from multicellular organisms to accomplish differential gene expression. It allows specialization of cells, and instead of lumping all cells together, you have cells doing different functions. Level 3, post-transcription. Many options result in many choices. Following transcription is 5-capping and 3-polyadenylation, and those are necessary for eukaryotic mRNA to remain functional and be transported to cytoplasm for translation. The reason there are so many options and so many choices is because alternative splicing. Alternative splicing of exons allows for multiple genes to be produced from a single transcript. For example, this transcript from 1, 2, 3, 4, 5 can be transcribed as 1, 2, 3, 5 or 1, 2, 4, 5. Number 4, pre-translation. Research has shown there's a lot of non-coding RNA, ncRNA, molecules in cells. RNA interference, or RNAi, are controlled by a group of small molecules, also known as microRNA or miRNA. The mRNA are produced after the transcript for them is cleaved into multiple fragments by disoproteins. Disoproteins are essential to RNA interference. Any mRNA with a sequence complementary to miRNA is tagged with the miRNA protein complex, and these tagged mRNA molecules are not translated. Level 5. Post-translation. Ubiquitin, which is a protein that is very abundant, will tag unnecessary proteins for transport to a proteasome. In the proteasome, it's either going to be broken down, or if it's an amino acid, it's going to be reusable. To make it easier to remember, you can view a proteasome as the trash can where leftovers go to be broken down or recycled. Why do eukaryotes have more levels of regulation of their genome than prokaryotes do? Simple. It's because of the decoupling of transcription and translation, which allows for more control points.